Okay, so welcome to the SMALP Industry Club. It's great to have you all here today. We typically meet at this time. There's a few newcomers, so I thought maybe we might start with introductions. You can share any news if you wish. Just to kick off, I'll just say that, well, it's really a pleasure to run this meeting again. It's been a lot of fun. Our last meeting was in Paris, and we're also going to be planning our next meeting probably at the end of June. I will be suggesting a couple dates. I'll be in Paris again starting on the 15th of April. I'm looking to do a remote meeting, and we'll also be exploring the possibility of doing an in-person meeting in the autumn. And so I look forward to that discussion as well. Would anyone else like to say hello, introduce themselves, and share any news that they wish?
Well, hi, this is Barry Bruce in Knoxville. We are we're pretty excited that one of the new polymers that we designed here and put in our Agavante Chem paper, I guess 2 years ago, we've now been able to get an extremely high resolution cryo-EM structure of that of a SMALP, and I think it'll probably be the world's largest SMALP structure, a little over 1.8 megadaltons. But that, we're actually getting we're under 2.1 angstrom resolution. We're pretty excited about that because this is a polymer that has been designed to maintain an oligomeric state that almost all other polymers disrupt. So we're excited about that. And then we have some new polymers that I think a.rothnie will talk about that appear to be, we're sort of discovering this opposing tendency. So my membranes have galactolipids, which are quite polar, and then there are a lot of phospholipid-rich membranes that have often much more saturated fatty acids, cholesterol, sphingolipids. And we find that polymers that work well in our case typically don't work well in mitochondria and vice versa. So it may be that we're starting to really come to some clarity on the structure-function relationship between polymer design and lipid profile of target membranes.
That's great, Bruce. Very exciting news.
And we're also trying to run them. We're thinking, and what people can talk about, we're thinking about doing the native nanomembrane protein #2 in the fall here in Knoxville, but I want to get feedback. There seems to be a lot of guarded concern about conferences in general,
yeah,
especially here in the United States.
Yeah, good points, Barry. Any questions for Barry? A lot of interesting updates there.
Barry, I do have a question. I think I missed the part. So what is the name for your polymer?
Well, we have a lot of, I work with Brian Long, who's a polymer chemist. And we have, I would guess we've designed in some over 60, maybe over 100 polymers. But these are what we call the ones that are working best in this particular application for the cryo-EM, are the ones that were in our Agavante Chem, which we often call the alpha MAs. These have a relatively variable length olefin group. And that seems to, I'm not sure, the styrene, has its ability to insert into the bilayers, but the olefins, I think, insert possibly in a completely different geometry. We don't really know, but I have a feeling the plane of the styrene and the axis of the olefin might be at right angles to each other, but that's only a speculation.
Okay, great. Thank you. Thanks.
Very exciting. I think we'll be interested to know if you'll be making those available collaboratively or commercially in the near future as well. So
I would say that we have had, we patented 3 of these things, and we are finally getting some licensing agreements with companies to put these in the marketplace. So I think that within 6 months, I hope you'll be able to purchase these things.
Okay.
great.
So that's really interesting because in my talk later, I'm going to include a little bit of data with some of those polymers too.
Yeah, I know. I alluded to that. I wasn't sure you were, but
Well, that's great news. Great to see the collaborations. Any other questions for Barry? Okay.
any of you who did attend the meeting in Knoxville, it would be nice to get your feedback. Michael and I have discussed it, and he feels that it was probably too long of a meeting. I don't know. It was 2.5 days. It was a pretty packed 2.5 days, but again, it does require traveling. But one of the attractive features of Knoxville is it's about 30% the cost of a major metropolitan area like New York or Boston or someplace like that.
Okay, Barry, and just to clarify, no one else has proposed to run a meeting, in-person meeting next autumn. So the floor is going to be yours on that one. And I mean, Birmingham also hosts great workshops. Alice can tell us about that and what her plans are, perhaps. Alice, do you want to chip in and say how things are with you?
So we're doing our membrane training workshop again in a couple of weeks in Birmingham. That's mainly aimed for people like new to the field of membrane proteins. And then we're going to be doing a general membrane protein conference in Birmingham next East, like 2026 Easter time. But that's more general membrane proteins rather than just SMALPs.
And that's with the Biochemical Society?
Yeah.
Okay. And we might be able to work with you on that one as well, in terms of like maybe doing the virtual piece for both. That's pretty easy, but I think it is increasingly difficult to get people to travel transcontinentally. So let's talk about that in a minute, maybe. While we're on the topic, Bert, do you want to, or Justel, do you want to talk about your any updates from you and the possibility of maybe a conference in 2027?
We were actually thinking 2026
Okay.
because our Welcome Trust grant runs until beginning of 2027, and we thought of sort of that as towards the end of the grant, but not beyond the end of the grant, if that makes sense.
Okay. Well, that's great. I think let's explore that, because actually I'm flexible in terms of, we may not be able to all get to both locations, but it might be possible to run a virtual part of it. If you're doing hybrid, that might work really well, or have like split virtual and in person. But we should talk about dates just to make sure there's complementarity. Maybe we can cover that in a few minutes. Anything else from South Africa?
And maybe Giselle wants to say a few words there.
I just wanted to say that Lauren Ball, who finished her postdoc with us last year and did a whole academic career in our lab, she's going to talk about some new developments in terms of blocker polymers with enhanced stability towards solution properties, in particular divalent cations, which seem to be very promising. So she'll be talking about that. And we're also using that to include some new functionalities on that blocker polymer system, which we're hoping to get tested. So if anyone's keen to collaborate or keen to try out some new materials, we're constantly developing some new polymers. So always open for a new partnership.
That's very helpful. Thank you so much. Any questions for Justel? I'm just wondering about commercialization and the availability to the general research community.
Yes, so as I hope you guys know, we have a startup company called NanoScene, which is a commercialization platform for innovative polymers that we're designing, particularly for membrane protein research and beyond. But the first step, of course, before commercializing anything is validating the technology and having as many test beds as possible. So we're always keen for people to try it out on different targets, different expression systems, to really see what the value of the new formulations are.
Great.
So yeah, from now we have the first commercial polymers known as the BZAM polymers. They're sold by Cube, called SMALP BZ, which is a unique tunable series where we incrementally modify hydrophobicity, and we're seeing very interesting results that is affected by these small changes in hydrophobicity on the actual polymer and its solubilization performance, but also its subsequent solution properties.
Great. And just to remind everybody, please do send me any publications or patents, and I will then put that on the library so people can be aware of those. And I do that every newsletter. I think there's also a place where you can upload that to a Google form. You'll see that in the emails. I wanted to thank also Krish for joining. They've got new products over at Anatrace. Any updates or news from Anatrace?
Hey, good morning, everyone. It's nice to meet you all today. We have a new product launched by Anatrace in US, and then Molecular Dimensions, which is our sister company in UK. It's a lipid screen with different combinations, 31 combinations of individual lipids and bi and trilipid mixes in triplicates. So these are available on the website right now, and they're already listed on the SMALP website as well. And I can quickly, if it's okay, I can take the screen and quickly share a couple of things on about the polymer, or sorry, on the lipid screen.
Sure. Just to tell you, Chris, your microphone is a little bit difficult to hear. And so maybe you
Okay.
want to check your mic or switch your mics, and then maybe we
Sure.
can go through that in a moment. Thank you for that very much. Any other news that people want to share at this point before we go into the regular agenda?
Can I just,
Yeah,
Yep.
can I,
yeah, Barry, go ahead. Yeah.
Gastel, I just wanted to say that we've tested all of your BZ series on bovine mitochondrial intermembrane membranes, and we have very good success isolating very large and multi-component respirosomes.
Oh,
So
fantastic.
if anyone else has tried that with mitochondrial intermembranes, but we are trying to finish that up, and so at least 2 of your polymers will be featured in that paper. So
That's
it looks
really
like they're
exciting.
very
Thank you
good,
so much
and
for sharing.
they're quite interesting in that, you know, there's 5 complexes in the membrane, and we're able to capture in discrete particles complex 2 and 4 and 3 and 5, and you know the BZ polymers seem to have really good success in preserving those native ensembles.
Fantastic. Thank you so much for the feedback. That's exciting.
Okay.
Love to see the research really soon.
Okay, great news. Any other news that people might want to share? Happy
Yeah.
to have our.
Yeah, Michael. Yeah,
Yeah, please.
maybe I can quickly share some like news from our company. So basically, we are from a different angle. I mean, just I heard talk about a lot of on the polymer side. So we are more focused on the biology, on the protein purification. So we build up our internal capacity like baculovirus and also stable cell line for the membrane protein production, because in the past membrane protein yield is relatively low, and so we boost up the production by using all different technology. And so we are happy to try all different polymers. If you guys have all different new polymer technology, we can try. And we also have the assay test platform, so we can evaluate whether this different platform polymer really can preserve the functionality of the protein. So if you do have a new polymer, our company are willing to test them and give you the data feedback. And in the future, if you would like to collaborate in commercially, we do also open for this opportunity. Yeah.
Great. Thank you so much. Any questions? I'm just wondering about the minimum amount that you need for a polymer and the kind of purity that you need to run it through your screen.
Yeah, the minimum amount, well, is depend on different polymer, right? So, Michael, if possible, for this new polymer, so any new innovation, could you share some of the like how we can acquire them? I mean, what is the company's name so we can buy from them? So in those information, if you could share in the group, then that would be helpful for us also. Yeah.
Yeah, I think there's a lot going on here. And one thing I might do, I do have a page of polymers that are commercially available, but maybe what I can do is set up an experimental polymer page where, like, if some of these polymers that are still being patented and not, maybe they'll be commercially available in 6 months or so. Maybe I can put those on as well. It would be nice to have like a link, let's say, so people could then see either your lab-based polymer, some quality metrics, like, ideally there's some whatever NMR, mass spec, FTIR, whatever, so that people know what they're getting. Because I think we want to make sure, and Barry has been pretty clear about this, that we need to maintain quality of polymers and also make sure we understand the stability. So, assuming that they meet those criteria, and maybe we need to set those criteria for like pre-commercial polymers, so that then people can just contact the source and get polymer and maybe get compensated for the cost. Like it might be that it costs a couple $1,000s to make a prep, and then we can make it a little more sustainable that way as well, so that people can get experimental polymer, but at a reasonable price and quality. Is that all right if we maybe explore that? So the teams that are here, if you have a polymer, send me a link and anything you want to tell us about the structure, um, and the quality or whatever. Maybe you have a PDF or something like a technical sheet, and then that can then be made available either collaboratively or at a cost. We also are developing polymers. We have a paper that's in press. Well, I mean, it's actually in submission on a series of polymers that we worked on with the team from South Africa. So Gestel in particular was key in that whole project. So I think it sounds like there's about 10 or so polymers coming down the pipeline in the next year. And it would be maybe ripe for us to figure out how to make that available maybe as a let's say at the prototype stage before commercial release. Would that be helpful?
Michael, I see this maybe having there's sort of two ways to skin this cat. One would be a repository where all of these polymers are provided and sent out to all these different labs to test it. I think that's probably a much more laborious and expensive thing.
Yep.
The other way is that maybe we would have a repository of target membranes and we would send those out to the different people. It's complicated with no support for technical staff to do really either one of these things.
Yeah, I mean, we could become a communications portal. That's easy. In terms of warehousing material, I think that's because it's IP and labor intensive. I think that's got to be whoever, made the polymers or is, that university or lab has to take responsibility for that and the relationship management as well. I suspect because people are at the end of the day looking for a commercial relationship to make it all sustainable. So my recommendation would be that each of us who is in the process of patenting and planning to make available commercial polymers, think about, you may not want 100 customers, but if you want, let's say 10 customers or so just to try. And I think what DEMA is doing is fantastic, and Cube and the other companies as well in terms of testing a large number of targets. So I think it makes sense to me that we could probably manage this without too much effort. The hard part is just making, you have to make 10-gram quantities probably of these polymers. That's a lot of work. And then you want a couple big clients to test out the early stage capacity and make sure it's good for commercial release. And then hopefully within a few months or whatever, you can then release. Would that make sense? Or do people think we should be warehousing either polymers or targets? Because I think I've got concerns that might be, we don't have the infrastructure.
From an academic synthesis lab perspective, 10 grams is really a lot of work. That would be really, that we work on graduate students. That would occupy a graduate student quite a bit of time, and they would see very little fruit from that, I think.
Yeah, but if you're going to commercialize, you're going to need
the right way,
Yeah. Well, maybe I can ask the Nanocene crowd, well, what's your approach to this? Do you primarily just provide the recipe, or do you provide the quantity that Cube needs, for example, for sales globally?
So, like we said, looking for partners for essentially beta testing the technology. We have a strong partnership with University of Birmingham, Tim Daffon. They're part of the Wellcome Trust grant. So the structure is we design something, they give it a first go, positive results, and then we seek an alternative or additional collaborator for the beta testing on different targets. So for that, we generally use like a couple 100 milligrams per testing round to see the viability, because like I said, it's obviously expensive and it takes a lot of time. But that would be for a new formulation and validating a new formulation. What we're doing at Nanocene now, which is extremely exciting, and we've been working on this for the last year. We've raised some funding at the end of last year, and we are piloting a a scale-up production of raft-made polymers, which is commenced to start in May. And we're using a manufacturing facility. So this would be the first time globally that this kind of chemistry would be attempted on this scale. So fingers crossed. We're very excited, also very nervous to see what's going to happen. But if we can make this all work, then we are creating a platform for the scaling of useful materials for membrane protein research. So yeah, please keep an eye. We will let you guys know how it goes. Hopefully in the next couple of months, we'll get some positive results.
That's great news. Is that based in Stellenbosch?
Yeah, it's based in South Africa. It's not in Stellenbosch, but it's in another province, so another region of South Africa. So it's a big week moving up there for a couple of weeks. We're going to do a tech transfer, see what it looks like on the scaled systems, and then yeah, hopefully everything goes accordingly.
That's great news.
Oops.
Congratulations. I know that's a huge investment of time and money, and certainly effort. In terms of is it mainly raft or is it going to also be batch?
So it will be raft made. This would be batch wise, but we'll also have some projects working on continuous like flow reactors to see whether we can also get that going.
So
Okay.
Gustavo, is this totally linked to your company, or is this a standalone facility that other chemists could contract to make larger quantities?
In this particular case, this is linked to the company. They mainly focus on pharmaceutical development. So this would be the first time for them as well doing something like this. So we're on both sides. We're piloting this kind of technology and this kind of chemistry. So yeah, we'll hopefully give you a good update soon.
That's great news. You probably know that in Johannesburg, there was also some reactors at the National Center there. I don't know if they're using them, but there was a lot of capacity there in Johannesburg, and I could put you in touch with the people there. I toured it maybe, I don't know, 6 years ago or so, and thought, wow, a lot of capacity, but not really well used.
Yeah, 100%. It would always great. We actually struggled a lot for someone who is willing to try this kind of chemistry because, as you know, raft is small molecule, and then you're transferring that to big molecule, and there's multiple synthetic steps in the making of the BZAM polymers. That's the first ones that we are piloting. So in terms of labor, it is a lot, and there's also multiple steps. So it's not just one pot system.
Great.
Yeah.
I just wanted to maybe introduce the people who are developing the equipment that could benefit. Malvern Panalytical in particular has got great equipment, a whole suite and new systems coming online. Is there a way for manufacturers of equipment to test the polymers and the discs for screening, binding kinetics, et cetera? Any questions maybe from Malvern Panalytical? Or any offers? Like I think probably some of these labs that are present here do have Malvern equipment. Like we have a ITC, for example, and a Zeta Sizer Ultra, very happy with them. And I think we're happy just to continue to publish with our user community on these systems. Are there any other issues about equipment and compatibility? Higher throughput, I guess, in terms of the Birmingham community, they're doing more high throughput, I guess, screening technology development. I think one of the questions is going to be screening, whether it's for antibodies or for small molecules binding to membrane targets. Does anyone want to speak to that? Okay.
Can I insert something?
Please.
Yeah, so my lab also develop polymers, and we mainly focus on hydrogen structure determination. So we have a platform to test all the polymers, especially we focus on the quality. So there's a lot of public polymer, they can purify protein, also claim get the lipids, but didn't show hydrogen structure determination. I think the quality is the issue, especially for pharmaceutical industry. For structure-based drug design or screening drug, they want to make sure the polymer purified membrane proteins in functional state and also suitable for hydrogen structure determination, especially showing the density. Use EM, you want to show the lipid density. Okay, so our lab have a platform to check because we develop a lot of polymer, like 100 of polymer, maybe 10 of them work really good. A lot of are trash, we would think. Not trashy, but based on accumulation of experience. But I think have a good polymer quality is a key thing, because if you market it, but it's not in good quality, people buy it, they try it, it didn't work. Then it said, oh, this polymer technology didn't work. I heard from some of the peers, they said, for example, I'm reviewing some papers published in Current Opinion Structural Biology. I was invited also too. So some people say that the polymer science currently is didn't work. We didn't see much access. There's not too many hydrogen structure published. Many still from detergent. I will argue that my lab we already solved more than 20 different hydrogens. We have not published yet. We have some patent we are planned, but we were able to solve structure from bacteria to human, even insect, plant. So, but we need to prove to the community that our polymer is in high quality, and we have good polymer. Then people will use that, and the pharmaceutical company will use our technology for drug discovery. That's very important, especially for GPCR, for example, is highly interested, but so far there's no successful GPCR structure determination with any of the polymers. So we already got some pretty good preliminary data, but we will need we have not published yet. Yeah, I also form a company. So if anyone want to us to test your polymer, we can test that. If that's good, we can also license to our company. I'm trying to license of my own polymer from our university to my company. So we call our company NCM Tech. So yeah, so we focus on the high quality and structure determination and functional validation about this technology. And I also, based on my experience, I also think that the polymer is not just the polymer itself, is the whole integrated technology. For example, there's some polymer you give to someone. I have a lot of collaborators. I send the polymer to some people. Some people, they got success, but someone, they didn't get success. Even told them how to do that, they still didn't get success. But for example, one collaborate, I sent the polymer to them, they didn't get success. They said, just come to my lab, ask your student come here. Then he can't get, I tell them exactly the thing, it didn't work. You know, it's not just a polymer, it also need experience. And sometimes the hands-on experience is also crucial, because polymer is kind of tricky, like a not like a detergent, so straightforward. But
So
I
those
think,
yeah.
we have a lot of expertise, like from Bert and Barry Bruce. You also develop a lot of polymers. I think really you need to rigorously test with membrane protein, different membrane. As Bruce said, so some polymers good for some membrane protein, but not good for other protein. So it's clearly we need a larger library if we want to target all the membrane protein. It's not one fit for all, definitely. Yeah. So need a collaborative effort.
So Michael, I have one, it's not maybe new news to y'all, but so we do, after we get our SMALPs, we'd like to do careful lipidomics. And so in the US, we've been going to Kansas State, Ruth Whitehall's group, very, very nice lab for doing quantitative lipidomics. But there's a lab that does the same thing at University of Bonn that maybe you people in Europe would want to use, that are Peter Dorman's group. Very good place for doing quantitative lipidomics. Again, there's a fee, but the pricing between the two, they do basically the same thing on two sides of the ocean. And it looks like the prices are pretty comparable if you want to do it. And I can tell you that the Kansas State group is got a pretty big backlog, because they are one of the few good academic labs that will do this stuff for a price. We have had struggles not once, but no, once, 3 times with other small molecule mass spec labs getting reliable quantitative lipidomics. So we're very happy with the Kansas State group, and I'm going to give the group in Germany a try as well. I can give you
Okay.
a link if people want.
Yeah, do share those links, Barry, and I can also push those out through the news. I think the issue about quality is really key, and I think one of the things this committee may want to do is decide what quality metrics are appropriate. Probably both, let's say, a Cube and a Trace and maybe other companies will want is technical data on quality. And I'm assuming they want C13, FTIR, maybe mass spec. Some of the things that we're looking to measure is, well, I guess the purity for sure. Also the PDI, just the chemical integrity and stability by NMR typically we do. So I think all of these are kind of maybe obvious to us, but if we can communicate that also to the researchers and to the companies that are using the polymers, maybe we can kind of set a bar for quality. The issue about functional effects, I think, is going to be variable, and I think GPCRs in particular are sensitive to polymers, maybe also photosystems that depend on particular lipids, galactolipids included. So I think the functional effects are maybe a little harder to characterize unless we do work with DEMA and Cube and the rest who can compare maybe hundreds of targets. So I think we have that capacity in this community here. So that's great. I want to turn it over to our colleagues from Malvern Panalytical. Would you like to say anything about your technology and maybe work with GPCRs, yours or your clients?
Sure. My name's Joanne von Graefe. I'm from Malvern Panalytical in Switzerland. Do you hear me okay?
Yep.
Okay, good stuff. I didn't prepare a presentation or something for you this afternoon, but I'm glad to tell you 5 minutes what we can do with our grating coupled interferometry. For anybody who hasn't heard of grating coupled interferometry, it's an alternative to surface plasmon resonance or biolayer interferometry. And one of the things that we're getting to be well known for, we have a good reputation for, is binding affinity determination with membrane proteins. Also with crude samples, we have some very well-known CROs working with our technology. Evotech is one, and they've been working on GPCR screening, fragment screening. We've been working with LeadX Pro in Switzerland, with TuBind in Germany. You keep mentioning the name Cube. They're testing our equipment right now. So, and we're pretty good at these membrane proteins.
That's fantastic. That's great news. And I think the research community may not be as aware as they could be. So I have put the information on the website about your LinkedIn note. Any other news about it or papers we can highlight, just let me know and we'll push it out. And I think, maybe getting papers out there that's going to build and if we can test our polymers against this. Is there a beta test lab where people can try the system?
There are multiple. We've got one in Switzerland, we've got one in France, we've got one in the UK, we've got one in the US.
Okay, if you'll give me a list of those, like maybe just the links to how people can access those, I can make that available if those labs are comfortable with that and share it with our
Sure.
community. We have
I mean,
about,
maybe the easiest way is I give you my email address, you contact me, and I'll let you know who you should contact.
yeah. You just
Good.
send me any email content and I'll put it up there and also link up with your colleagues. Perfect.
Leah, can I ask you a question? You Melbourne also markets, is it called the nanosite or the eyesight?
Nanosite.
I can tell you that
Nanosite.
we have had very good luck with that. Much better luck with that than the mass photometer that is a different technology, but is in a sense a competitor. And we don't fully have a good answer for that. People have told me that the problem with the SMALPs is that their negative charge is repellent and it changes the the particle classifications in the mass photometer, but we had great results with that with your instrument. But it's not your it's your interferometer based on Daryl Bornap's technology out of Vanderbilt?
No,
Backscattering?
no.
Okay. Different backs. Okay.
I do.
Yes.
I know the person that represents Nanosite Pro well, though, so I'll pass that on. They may want to reach out if that's okay.
Okay.
Yeah, thank you for sharing.
Thank you all very much for sharing. And actually, nobody does a formal presentation here. This is just a friendly roundtable. And the idea is we just share information. And it's a private meeting. I do share the video so you can check what somebody said if there's any questions or connect with them. So it's just basically a friendly roundtable here. And if you do want to do a presentation, you're welcome to either ask for a slot at a future conference or maybe have a user who can talk about their latest data. GPCR screening, of course, would be of central interest to this community. Okay,
Good to know. Thank you.
great. Any other? I just want to say hello to Rich Gandor as well. If you want to say anything, Rich, to the crowd, feel free. You've also been developing polymers at Virginia Tech.
Yeah, hi. I was late getting in, and I ended up using DuckDuckGo as my web browser, which blocks the video. So it's great for when I want to buy something, but it's not great for communication. As maybe you don't know, but everybody doesn't know, but a former student of ours is now working in the federal government and is continuing the project of making our still being copolymers, maleic acid, maleic anhydride copolymers. And that's been a slow process, but I think she's finally, at least in the latest communication, said that she's going to have some things ready for testing, which will be good. And of course, we don't know for sure, but we believe that these precisely alternating polymers will have higher quality than other things that are more random. But that's yet to be determined. So I think she's been in contact with you, Michael. So you probably know a little bit more than I do about what she's going on, because she has a formal communication with you, and because she works for the government, she can't. I don't have all the information of what she's doing. So maybe you could fill people in on what you know from Chanel.
Sure. So Chanel has published in the area, and I think there is an additional patent, at least that was filed a little while ago. So I think they're getting ready to be ready to talk about it. She is working for government, so she has to be careful in terms of what she says, but she is happy to talk about it as soon as she can. And I think one of the opportunities might be for her to talk next autumn if we have a meeting, for example, in Knoxville. I think she'd be happy to give a talk there, Barry. She's given talks before at this conference, and so people will probably know her already. So yeah, I think that could be really useful. We might want to talk about conferences now. I want to keep this meeting to an hour if possible, just because I know you're all busy. So is there anything else before we dive into the discussion of the next meeting and conference dates? I did want to maybe break it up into a couple things. Maybe just do it by time series. We do want to have a meeting in Boston. That's been brought up several times. Dima is going to be at a conference in Boston, I guess, is it May the 10th? Is that right?
Yeah, we do have, we are attending two conferences in Boston. One is Chinese Antibody Society annual meeting. Another one is PAX meeting, PAX Boston meeting, and it's also the annual meeting. So it's in May 10th to May 17th. Yeah.
Okay. Do you think it makes any sense for us to have a virtual meeting as a part of that? It could be a satellite or kind of a sister meeting where we have, it could just be an hour, it could be 3 hours, where we just have, like we're doing today, maybe having more industry focused talks. I think what we really want to do at some point is have an industry conference as well in Boston, possibly also the Bay Area. Could also be other places, but Boston has been the #1 destination for that meeting. I haven't quite been able to organize a physical meeting there, but a virtual we could definitely do. What's your thought about having, let's just say, make it easy, 3 talks, 1 hour, for a Boston meeting in that May week?
Yeah, I think a Boston place is definitely the most innovative place. You have the best university in the area, and also all the pharmaceutical company has a branch there. And I think it's you have a lot of interest people on the GPCR nanodisk technology. I mean, at this stage, when we talk with our biopharma customers, a lot of people still um, didn't heard about this nanodisk technology. They are still, thinking about the detergent approach. I think a nanodisk is definitely a way to go. I mean, for functional proteins, yeah.
Okay. Well, if you hear that they would be interested in having kind of a joint meeting, like ideally we would be able to um, market to especially the pharmaceutical sector clients. It doesn't need to be big. Like it might be just like 50 or so pharma people. It could be more.
Yeah.
If it's virtual, it can be as big as possible. Just let me know if you're
Yeah.
willing to ask the organizers, or if you want to do it together, we could just do a DEMA sponsored, like a 1 hour, whatever. If there's a lunch slot or something, we can just do, 3 lunch kind of talks, 20 minutes, and then
Sure, no
profile
problem.
maybe some of the new polymers that are coming out from
Yeah.
these spinouts here. They might be interested to know not just that, Cube is doing this because they probably know Cube already, but the
Yeah.
nanoscenes, the other small companies that are actively developing new polymers and are
Yeah.
really at the cutting edge. Okay.
Yeah.
Cool.
Yeah, I think right now, from my understanding, from my communication with the biopharma customers, right now the one of the main, advantage. I mean, one of the main progress in the drug development is the AI medicine. So for the new small molecule drug and antibody-based drug, the AI design medicine on this structure-based. It is one of the key, with the cryo-EM and for all this membrane protein with the polymer, I mean, get in. So you don't need like previously you do crystallography, you need a milligram of protein, but you just need a microgram level. You can get a high resolution structure, and this will be really speed up on the drug development. In the past, when you screen large, like millions of different molecules to find the best drug. Right now you just need with the AI design structure-based. They call it SBDDD, this technology. So you can just narrow down hundreds of them. You can find the best molecule. Inhibitor or the drug. So I think this in the future this will be a lot of progress in this field. I mean, nanodisk polymers will have a lot of future in this direction. Yeah.
Okay, thank you. I agree with all of that. And thank you also, Chris, for sharing. I will share the chat space and I will extract that if there's any public news as well to the community. So I think.
Sure. Thank you.
All right, great. So let's continue with that conversation offline and see if we can do something for the May 7th to 10th kind of period. Then in terms of the next SMALP conference, or what I call now Membrane Protein Conference, I've got 4 dates that I want to pass by you. So that's June 20th, 24th, 25th, and 26th. So usually what I do is ask here, and then if anyone says one of those dates don't work, let me know. Like I'll usually propose, let's say 2 or 3, and then that will be decided after the last talk. So if
Michael, is this your one-day Paris meeting, or is this a online?
No, this is all online. I mean, I might have lunch with people or dinner or something.
Do you have another one-day Paris meeting scheduled?
No.
Okay.
No, like I think I'll do it if Knoxville doesn't work out.
Yeah,
But
people should tell me. I mean, we have the space. It's essentially the first week of October, but I need some enthusiastic response. Now with the, we pissed off almost everybody in the world, including Michael's country. People might not want to come to the US. So this doesn't make my job any easier.
well, I think this is a real challenge. So let's just do one at a time. So in June, we'll have our regular quarterly Zoom meeting, and it'll be like, let's say 3 hours, same format. We'll have also have an industry club meeting beforehand. I'll be in Paris. If people want to meet up for lunch or something on that day in Paris, that's great. I'm happy to have a chat with anyone who wants to. But I'm just at this point focusing on trying to get a date you're happy with for the June meeting. You could just write in the chat space if you don't like those dates. Otherwise, I'm going to propose them in about 5 hours or so, 4 hours. Okay, then let's go to October. You're saying the first week of October, but I think it'll be contingent on getting some funding, right? Because at the end of the day, to do it in person, you're going to be looking for on the order of 10K or more, probably minimally in sponsorship, correct?
Last year we uh, raised about 12K and we actually had some funds left over. So I would say, between 8 and 12K of sponsorship would keep it. I mean, the main price savings, if you want to call it, is that hotels can be had for 125 to 130 a night, versus 280 to 300 in most big cities. So that's not really that's a cost that is extended to the participants. And then using a university conference facility keeps it really nominal fees. So I don't know what we charged last year, but we wouldn't probably have to increase the registration at all.
Yeah, I think it was about 200, 400, and 600 for students, faculty, and industry. So we can do the same format. I think what Barry is saying is he needs on the order of 8K. So if any sponsors want to support an in-person event, I'll circulate that as well. Then that'll be a go-ahead. If we don't get the money, let's say 2 or 3 months beforehand, then we'll just do another virtual, possibly also with a component in person in Paris. Okay.
Yep. Good.
So Barry, I'll leave that with you and the sponsors to decide if they are willing to commit. We can help as much as we can on our network, but we don't have the funding within our current budget to provide that within the SMALP network. So we do that as an additional, like the in-person is an additional sponsorship, let's say, on the same order of what you already give if you're a sponsor for that in-person. So, okay, so we got some information on the date. So 24th or 25th, which is, I think, a Tuesday or Wednesday, are looking preferable. So that's helpful. If anyone else has any concerns about the 24th or 25th, let me know. Then let's turn to the meeting in South Africa. What are your thoughts in terms of possible dates?
So we have a meeting this year, polymer meeting, on polymer technology itself, not related to SMALPs at all. And that is in the week of 9 to 12 November of this year. And we were thinking a similar time next year could work for us.
Okay, so around second week of November 2026?
Yep.
Yeah.
So
okay.
that is sort of 1 week, bit more than a week before Thanksgiving, which is clearly an important thing on the US side of the world.
Yeah. Okay. Any concerns about that? I think that'd be fantastic. I think
Bert, would this be in Johannesburg or where?
So the one that we organized this year is in Stellenbosch itself, which is one of the options that we have. Gestel came up with the idea. We have in the past also done conferences inside the Kruger National Park in one of the rest camps. That would make it more expensive. But since many people that come to South Africa want to go there anyway, that could be an option. But that's something that we'll have to sort of hear from potential attendees.
Well, they would both be wonderful.
Yeah, I think that'd be great. Those both sound
We're
fantastic.
excited, so hopefully you're too.
Why don't we discuss that briefly in the conference and see what the interest is? There might be some sponsors that really like the idea who aren't in this meeting here. And I think if we get some sponsorship, that will help with student bursaries or whatever. For that. Okay, sounds good. And then Birmingham, a meeting in Birmingham. I'm not sure, Alice, if you're able to speak again to the time frame of the 2026 meeting.
Yeah, it's around Easter. I can't remember the precise dates off the top of my head. I've got them somewhere.
Okay.
I think let me just have a
That's at
look.
Aston?
It's in central Birmingham. It's at a conference location in central Birmingham.
Okay. So I think that would be fine because they're kind of spaced apart, right?
Yeah.
They're not really conflicting in terms of time. So, and I think we may need to explore either hybrid meetings, given the travel situation. I think we're headed into kind of a difficult economic period, frankly, in terms of funding in the US and possibly elsewhere. So I think I would caution people just to think flexibly about, like in Paris, what we did is we did a virtual day and then we did in person. And that way people could just come for the day for the in-person and also just come for the virtual. So that reduced costs and increased flexibility. A hybrid event is much more expensive and complicated to run, but if people are willing to do that, that's another option. So I'll leave that to you to think about. I think in Kruger National Park, that's a great destination. I think we just have to see what the feeling would be about that. I mean, certainly a lot of people would love to go, but I think the costs are going to be significant, as you say, both for travel and the venue. So,
also, if you want to combine a hybrid part to it, then I think Kruger will not out-suspect. It's not a viable option. Whereas the Stellenbosch option is university-associated premises, and all the facilities are there for doing it hybrid. So that should not be a problem.
Yeah. So actually, what I find is the easiest is if we do on day one just virtual. And people pay a low cost for that, whatever, $50 or something. And then from day two onwards, it's in person. And then the cost may be a couple $100. But we split it that way. And that way you get the numbers on the virtual. And then the in person, you get the quality of the networking. And so that model works reasonably well in terms of the amount of effort and cost management. So why don't you think about whether or not that might work if you have one day in Stellenbosch and one day or 2 days or 3 days in Kruger, and then we can assess.
Doing it in two places is we talk significant distances here. I mean, South Africa is about the size of Western Europe. Yeah. That means you have to fly from Amsterdam to Athens. You know, a similar distance.
Mm-hmm. I don't think the virtual can be anywhere, right? So the virtual you can be in any place where you have network.
Sure. No. But what I mean to say is, you quickly said we do one day in Stellenbosch and then something in Kruger. I don't think that is an option. We either choose the one or the other location, and I think if we can do it similar to what Barry did last year in Knoxville, that was a great conference, and the great majority of it was in person. That's still my preference. Mm-hmm. So I think if we can sort of get a feel of how many people would be interested in coming to Stellenbosch, then we can start Stellenbosch or the Kruger National Park. We can start investigating and looking at the cost of both options.
Okay, I like that. I did want to ask people about, because we didn't do a formal debrief of the Paris event. So we had one day that basically I wasn't able to make it to the in-person because of a health issue, but I did the previous day on the Sunday, the virtual. And so that was a split conference. Did people have any thoughts on whether or not that worked for them? I thought one of the advantages was the virtual, you can get more people in the door. And then the in-person, you can get the quality of the in-person connectivity and networking. So that was quite manageable on a budget, basically. Any criticisms, comments about how that worked for the people
Michael,
here?
Do you think that, I mean, most of the ones that you manage, you have such low cost registration, there's really not much revenue raising, but if you raise the online costs significantly, it could be a method of underwriting the in-person cost.
Yeah, that's a good point. Now, people have wanted the events to be quite accessible, and I didn't want cost to be a barrier, particularly for students. So that's why the costs are low. It just kind of covers the very basic costs, not really people costs, but just like running Zoom and the mailer, that kind of thing. So I can certainly increase it, and I might increase it, let's say 10% or something, but I would be a little hesitant to double it, let's say.
I can just say that having run a lot of meetings hybrid, it increases the complexity in my mind and the stress of an organizer, because something undoubtedly doesn't work when you have an online component, and then you get behind schedule and the whole thing just collapses. You do it pretty well, Michael, but I've just not had it work well ever.
Yeah, I do a lot of conferences here as well, and we do them hybrid, and it is about 10 times the work and cost. So that's why I'm suggesting split, first Zoom one day and then in person the next 2 days or whatever.
Yeah, that's much more sensible than dovetailing them.
Yeah, and if you pay for the in-person, you get the virtual for free, and the virtual is low cost, basically. So you maximize impact and accessibility for like, let's say busy pharma or students who can't travel for family reasons or whatever. Right?
Bert and Castell, what's your time difference between Europe is what? Are y'all 1 hour?
It's now 1 hour, and next month it will be 0.
Okay. Yeah, I mean, that's a big advantage of, instead of going east-west, going north-south. So
correct.
You will be able to engage the European community without much problem.
Yeah. I mean, I think this this is a problem really across the world now. We need to have really cost-effective, accessible conferencing at a time when budgets are tight again. So I'm prepared to explore whatever works for people. If they want to do hybrid, fine. But I certainly wouldn't make it a requirement, and I would try and find a way to keep the costs under control as much as possible. I think even if I doubled the prices, we'd only generate maybe $2,000 or $3,000 more, and probably we'd lose some customers. So it's a little unpredictable. It wouldn't cover basically you need $10,000, I think, to run an in-person. So people have got to go. So any last comments before we close this meeting, the formal meeting? I want to thank you all for joining in. I will share the video and the chat, and I'll see you in an hour. Thank you all for chipping in and helping in so many different ways. And let's follow up on those conversations and see where we get with these possible conferences and funding mechanisms.
Michael, thank you.
Thank you so much.
Bye, everyone.
Cheers.
Cheers.